Right. Hi, everyone. I think maybe we can start soon. I think most of people are here. So yeah, I think we can go ahead. So good morning, everyone. And I would like to thank you all to attend today's presentation from Dr. Karim Mahidde. So Dr. Mahidde is a senior scientist from University of California, San Francisco, in the Department of Laboratory Medicine. Dr. Mahidde is a cancer immunologist that looks at the role of a tumor hypoxia in the immune response to a solid tumor. And so he's going to show today his work where he used respiratory hyperoxia to alleviate tumor hypoxia, and showing T-cell independent fashion to reduce tumor burden. So with that, I would like to give the floor to Dr. Mahidde, and we can start the presentation.
Okay, thank you, Vadim. So thank you everyone for being here, and thanks to giving me this opportunity to present our work consisting to study the role of tumor hypoxia in the control of immune response over the solid tumor development. So, as you probably know, blood, lymphatic vessels, fibroblasts, immune cells, chemical-physical components such as the extracellular matrix, the ECM, constitute component of the stromal compartment of the tumor microenvironment. The ability of the cells to interact with cancer cells will determine the clinical outcomes. As the tumor progresses, cancer and stromal cells undergo a restricted access in nutrient and oxygen. This phenomenon is referred as the tumor hypoxia, in which severity varies between different tumor types. Tumor hypoxia can stimulate a complex cell signaling network in cancer cells, including the PI3 kinase, the MAP kinase, the NF-kappa B, the hypoxia-inducing factor signaling pathway. The ability of all of the signaling pathway to interact each other will create positive and negative feedback loop to improve or trying to decrease the hypoxic effect. The tumor hypoxia acts as a driving force of tumor progression and metastasis. It can control directly the metastatic niche formation and tumor cell trafficking, but can also lead to a dysfunctional tumor vascularization by promoting vasculogenesis, angiogenesis, or can also control the acquisition of an epithelial to mesenchymal transition phenotype switch to promote tumor migration and metastasis. Tumor hypoxia can also control the cell cycle arrest, thus protecting from any external stress to confer radioresistance or chemoresistance by decreasing drug delivery within the tumor microenvironment or by altering their cytotoxic effect. Tumor hypoxia can also directly or indirectly influence the function of immune cells and the tumor development. For example, tumor hypoxia can directly inhibit T-cell response by altering proliferation, activation of T-cells, production of cytokines such as the granzyme B. And tumor hypoxia can also indirectly influence this response through interaction of T-cells, T-cells with tumor cells, for example, tumor microenvironment. Another possibility to inhibit the T-cell response is through interaction between T-cells and suppressive myeloid cells, the MDSCs, or tumor-associated macrophages. In that case, it has been shown that tumor hypoxia can promote the recruitment of PMN MDSCs, which result of a product of a deranged PMN differentiation program, leading to T-cell suppressive activity. This T-cell suppressive activity can be revealed in many ways. For example, the production by MDSCs of oxygen species can inhibit T-cell activation, proliferation by inhibiting TCR signaling, inhibiting the activity of the co-filling, the NF-kappa B pathway. Another possibility to inhibit this T-cell response is through the release of serine proteases such as the neutrophil elastase through the release of primary granules, which will inactivate cytokine in the tumor microenvironment, such as the IL-6, TNF-alpha, IL-2, or their corresponding receptor at the surface of T cells. Another well-described mechanism is the interaction between MDSCs with T cell through this interaction with PD-L1 and the exhaustion marker PD-1. To date, much of the work on tumor hypoxia on PMN and PMN MDSCs has been performed on isolated cells in vitro or through the use of mice with myeloid cell-targeted genetic disruption or genetic disruption of HIF pathway component. In vivo, aside of the use of antisense oligonucleotides or drug targeting the HIF signaling pathway, the therapeutic relief of tumor hypoxia has been shown to foster the anti-tumor T-cell response. However, in that case, the exact role of this PMN MDSCs was not clearly assessed. Another possibility was the use of drugs, for example, the use of the TH302 or evofosfamide, which will covalently bind the most severe hypoxic cells to impair DNA replication, leading to the apoptosis of these tumor epithelial cells. In that case, it has been shown that the use of this drug could improve the T-cell response, and this improved T-cell response was associated with a decrease in the frequency of PMN MDSCs. 15, 20 years ago, the use of the hyperbaric oxygen therapy, the HBOT, was also shown. This HBOT consists in a cycle between normoxia and hyperoxia at 90% oxygen, which is basically used in case of air or gas embolism, carbon monoxide poisoning, decompression sickness, etc. In the context of cancer, the use of this HBOT has been shown to reduce tumor growth on breast xenograft using SCID mice, so independently of T cells. It has also been shown a delay in tumor growth on mammary carcinoma in mice. Our goal in the lab is to understand how tumor hypoxia compromises innate and adaptive immune response cells to promote the tumor cell development. This is a complex signaling involving a triangular relationship between tumor hypoxia, T-cell response, as well as the tumor-associated PMNs, neutrophil, MDSCs. Given the role of this tumor-associated neutrophil MDSCs in tumorigenesis by limiting tumor cell killing, improving tumor cell proliferation, or generating an immunosuppressive microenvironment, we decided first to focus on this first axis between tumor hypoxia, between the relationship between tumor hypoxia and tumor-associated PMN. Adding complexity to this system, neutrophil, polymorphonuclear neutrophil, are thought to play an important but a controversial role in cancer. Indeed, in human, an elevated blood number of neutrophil in the blood, it's often associated with a poor prognosis in many kinds of human cancer. In mice, this increase in the blood, it's often associated with a tumor-promoting role of neutrophil by controlling initiation, migration, invasion, proliferation of tumor cells, by generating an immunosuppressive microenvironment, as shown before, or by promoting angiogenesis. In some cases, however, neutrophils are believed to support an anti-tumor role. For example, in lung cancer, neutrophil can foster the T-cell response. In the lab, we show in a model, in a mouse model recapitulating the human endometrioid adenocarcinoma that neutrophils were able to induce the tumor sloughing, which is the detachment of a live tumor cell from the basement membrane to oppose tumorigenesis. So what can explain this difference? First, different cancer can respond differently to PMNs. Second, the neutrophil can acquire a uniform specialized phenotype within the tumor microenvironment. For example, this is the case of the tumor-associated neutrophil 1 subset with a pro-inflammatory and anti-tumor functions, which can become tumor-associated neutrophil 2 pro-tumor in presence of TGF-beta in mouse model recapitulating melanoma. It's also the case for the granulocytic MDSCs, which are present in blood and tissue of human patients and in mice. And more recently, a PMN subset expressing Siglec-F has been shown to have pro-tumor properties, whereas another PMN subset with an HLA class 2 signature, the hybrid TAN, has been shown to foster the T-cell response. So adding more complexity to the system, different PMN-derived products can also have different effects in vivo. For example, the neutrophil elastase has been shown to contribute to the epidermal destruction in the context of the bullous pemphigoid disease, or can directly foster tumor cell proliferation in a model of lung cancer. This is also the case for the reactive oxygen species, the ROS, regardless from their source. ROS can have a mitochondria source or a source from the NADPH complex. This oxygen species has been shown to induce epithelial damage in the context of the inflammatory bowel disease, or can restrain the malignant process at the pulmonary metastatic site, or inhibit the adaptive immune response. So one question is to understand how tumor hypoxia can control not only the neutrophil, tumor-associated neutrophil trafficking, but also the fate as well as the functionality of neutrophil. In the lab, we use a mouse model recapitulating the human endometrioid adenocarcinoma. Endometrioid adenocarcinoma can be divided into subtype. The type 2, the serous form, which is the most aggressive form of uterine cancer, and the endometrioid form, or type 1, which is the most prevalent malignancy in women. In 50% of the case, this endometrioid adenocarcinoma involved the deletion of the tumor suppressor P10, leading to a constitutive phosphorylation of AKT, thus promoting tumorigenesis. So in the lab, we use a mouse model where the Cre recombinase is under the control of the progesterone receptor promoter, leading to a conditional deletion of the gene P10 in different organs such as the breast, ovary, uterus, leading to the tumorigenesis only in the endometrium. From these PR3 P10 locks, mice I will refer as the purple mice during this talk, and as opposed to the control mice, the PL, we found that this conditional deletion led to the constitutive phosphorylation of AKT, which reflect a massive proliferation of tumor epithelial cells as reflected by the staining of these epithelial cells by the ikaderine staining. From this mice, we show that these purple mice at 3 weeks of age exhibit a very structured structure with a massive and uniform hyperplasia compared to the control PL mice. At 4 weeks of age, so at early stage of the tumor development, the structure of the purple uteri appears to be a lot more disorganized, with a lot of intraluminal debris, as you can see here with the asterisks, and a massive infiltration of inflammatory leukocytes. And by flow cytometry, we determined that these leukocytes were neutrophil. As you can see here, neutrophil in green strongly colocalized with the tumor epithelial cells. So using depleting antibodies or through the use of the genetic disruption of the CSF3 receptor, which is the receptor of the GCSF, the growth factor of neutrophil. So these purple CSF3 mice don't have neutrophil. We find a massive increase in tumor burden compared to the regular purple mice. Moreover, we found a total absence in intraluminal debris. Moreover, this mice lacking neutrophil dies earlier than the regular purple mice. Here is an example of what uteri of a control mice, a purple and a purple mice without neutrophil looks like. We demonstrated here that the neutrophil were essential to restrain the tumor growth and the progression. Indeed, we found that this intraluminal debris corresponded in fact to live epithelial cells falling off from the basement membrane. Very importantly, we found that this tumor sloughing and the opposition to tumor growth on these mice at early stage of tumor development was totally T-cell independent. So going back now to tumor hypoxia. We found using this model that the PMN influx was strongly associated with tumor hypoxia. As you can see here, indeed, neutrophil in green strongly colocalized with the HIF1 alpha stabilization. Moreover, we also found that neutrophil also colocalized with the more hypoxic area, as you can see here with the staining with the pimonidazole. The pimonidazole, it's a chemical reagent which will covalently bind the more severe hypoxic cells and which will be revealed by immunohistochemistry. Importantly, we found that the tumor cell hypoxia was strongly associated with the induction of CXCR2 ligand, so chemoattractant of neutrophil. So CXCL1, CXCL2, and mostly CXCL5. So in sum, using this model recapitulating the human disease, we found that the hypoxia-induced inflammation induced the production of CXCL1, CXCL2, CXCL5 chemoattractant of neutrophil, leading to the PMN recruitment in situ, allowing the cells to induce the tumor cell detachment. So given that, we considered that this mouse model was a fantastic opportunity to trying to understand not only the connection between neutrophil, between a tumor hypoxia and neutrophil influx, but also the connection between tumor hypoxia and the PMN response independently of T cells. So for that, to assess the role of hypoxia in vivo on this PMN tumor cell interaction, we developed a strategy, the Hyperoxia condition, which consists to increase the oxygen delivery to the core of the tissue by increasing the partial pressure of oxygen dissolved in arterial blood. So for that, we housed mice in an environmental chamber for the last 10 days prior to sacrifice at 60% of oxygen. This condition will be referred as the Hyperoxia condition and compared to the normoxia condition where the mice are housed at ambient oxygen. Importantly, this system has been previously shown to improve tumor oxygenation in non-pregnant uterus. So as you can see here, we didn't find any sign of lung inflammation nor recruitment of pro-inflammatory leukocyte, which is consistent with a long-term exposure at this 60% oxygen observed in other species. So from these mice, we found that the hyperoxia reduces the PMN density in the tumor-bearing uterus by 50%. The PMN density corresponds to a number of cells per milligram of tissue. Importantly, the density of other leukocytes remained unchanged. Very importantly, this decrease in PMN density was not due to a defect in granulopoiesis induced by the hyperoxia. Indeed, we didn't find in the blood of hyperoxia purple mice a decrease in blood PMN. In fact, it's the opposite, and we find an accumulation of mature neutrophil in the blood of these mice. Consistent with this process, we found that hyperoxia reduced the production of CXCL5 by the epithelial tumor cells, which will reflect a defect in the PMN recruitment in the tumor-bearing uterus. So importantly, neutrophils are known to induce an inflammatory response and also to induce hypoxia through a feed-forward loop. So to make sure that this effect was not a de novo effect or due to this feed-forward loop, we performed the same hyperoxia experiment on mice lacking neutrophil. And what we found, similarly, on mice lacking neutrophil is the same decrease in CXCL5 production by epithelial cells upon relief of tumor hypoxia. Associated with this reduction, we found a massive reduction in HIF-1 alpha stabilization, more than 70% of average. And here is a representative picture. So this suggested that hyperoxia has a PMN-independent effect on purple tumor cells. So as I told you before, neutrophils are required to oppose tumorigenesis. So given that hyperoxia reduces the PMN density, we expected that hyperoxia will induce an increase in tumor burden. So very surprisingly, we found that hyperoxia reduces the tumor burden on purple mice by more than 50%, 60% in average. This phenomenon was independent of T cells since the housing of purple mice without T cells induced a similar reduction in tumor burden. By contrast, we didn't notice any difference in tumor burden while housing purple mice lacking neutrophil, suggesting that the hyperoxia reduces the purple tumor burden in a PMN-dependent fashion. As mentioned previously, the mechanism through which neutrophil oppose tumorigenesis is by inducing the sloughing of live epithelial tumor cells. This was associated with an increase in denuded endometrial area, which are area lacking epithelial cells. All of these phenomena were not apparent on mice lacking neutrophil. Consistent with this process, we also found that neutrophil induced the abrogation of the alpha-6 beta-4 complex, as you can see here, with the loss of alpha-6 staining, which is absent or discontinuous, whereas in absence of neutrophil, the staining is totally continuous. You probably know that this alpha-6 beta-4 complex is required to maintain a strong anchoring of epithelial cells to the basement membrane. So given that, we decided to evaluate these parameters of killing and how they were affected on purple mice upon relief of tumor hypoxia. As you can see here, relief of tumor hypoxia increased the percentage of intraluminal debris, as you can see here, and in the representative picture. This was associated with an increase in the percentage of denuded endometrial surface in hyperoxia condition. And consistent with this, we found a decrease in alpha-6 staining. Importantly, all of these parameters remain unchanged in purple mice lacking neutrophil upon relief of tumor hypoxia, suggesting that this improved killing was due only to neutrophil. Importantly, we found previously, if you look at the baseline, the black circle, that the tumor proliferation rate, which is revealed through the pH3 staining, was higher on purple mice compared to the purple mice lacking neutrophil. We talked at the beginning that this increased proliferation rate was due to a wound-healing response to tumor cell killing. But we found that relieving tumor hypoxia decreased the tumor proliferation rate on purple mice, but not on purple mice lacking neutrophil, suggesting that moreover than the killing properties of neutrophil, the cells were also able to induce tumor cell proliferation, a mechanism abrogated upon relief of tumor hypoxia. So as a first conclusion of this study, we found that the hypoxia can promote the recruitment to the purple promote the recruitment of neutrophil to the purple uterus, but will prevent the cells to reach their maximum combating potential in a two-pronged effect. First, by limiting the anti-tumor activity of neutrophil, and second, by directly promoting the tumor cell proliferation. So this is very interesting because we always thought that neutrophil in aggregate were either pro-tumor or anti-tumor cells. But in fact, what we show here is that each neutrophil share both pro-tumor or anti-tumor activity, and that the balance between this pro-tumor and anti-tumor activity are likely controlled by tumor hypoxia. The relief of tumor hypoxia will increase the PMN-dependent restraint of tumor growth, thus compensating the associated reduction in PMN density. So the next question was to determine which PMN-derived product may impact the tumor growth, and how does hyperoxia will affect this product. So for that, we generated purple mice with a genetic lesion on some PMN-derived products which are known to have a role in cancer. For example, we generated purple ELANE knockout mice, purple mice lacking neutrophil elastase activity. We generated purple CYBB deficient mice, where CYBB encodes for the GP91 FOX subunit of the NADPH oxidase complex. So on these mice, only the production of oxygen species will be deficient by the NOX2 complex, but not by the mitochondria. And we generated purple MMP9 knockout mice deficient for MMP9 activity. As you can see in the graph, housing of this mutant in hyperoxia condition didn't change the tumor burden while compared to the respective baseline. In the case of the neutrophil elastase, we found that the tumor burden of this mice under normoxia condition was lower in the purple elastase knockout mouse compared to the regular purple mouse. This reduction was due to a decreased tumor cell proliferation rate, which is consistent with its role previously described in lung cancer. Moreover, the hyperoxia housing condition of purple elastase knockout mice didn't change the tumor burden, as well as did not further decrease the tumor proliferation rate than the normoxia counterpart, suggesting that the neutrophil elastase fosters the tumor cell proliferation by limiting its influence. Consistent with this role, we found that the percentage of denuded endometrioid surface was still increased upon relief of tumor hypoxia on this mutant, whereas the alpha-6 staining was still decreased on purple elastase mice upon relief of tumor hypoxia, suggesting that neutrophil elastase don't play any role in the tumor cell killing. With a similar analysis, as I mentioned before, we found that the tumor burden on purple CYBB knockout or purple MMP9 knockout mice was not affected upon relief of tumor hypoxia. Consistent with this process, we found that the percentage of denuded area as well as the percentage of intraluminal debris remained unchanged while comparing the hyperoxia condition of each of these mutants with their respective baseline. Similarly, we didn't find any change in alpha-6 staining on purple MMP9 mice, and although there is a significant decrease in alpha-6 staining on purple CYBB mice, the baseline is so high compared to the normoxia baseline that we could conclude that there was a dual requirement of both oxygen species from the NOx2 complex and MMP9 involved in the tumor cell killing. So just keep in mind in this second conclusion that ROS production and MMP9 activity are both involved in tumor cell killing, whereas neutrophil elastase control the tumor cell proliferation. I won't show you the result here, but we also found that neutrophil elastase also controls the advance of severe hypoxia, so the generation of an immunosuppressive microenvironment. So now to get a greater insight about how tissue hypoxia alters the functionality of tumor-associated neutrophil, we sorted PMN from uteri of purple normoxia and hyperoxia mice to perform an RNA-seq. So among the 423 genes differentially expressed between these conditions, we found that CYBB encoding for the GP91 FOX subunit was increased, suggesting more ROS production. We also found that MMP2 and MMP14 metalloproteases, which are known to be activators of MMP9, were also up-regulated. MMP9, it's already highly transcribed in both normoxia and hyperoxia condition and remain unchanged. However, and consistent with this MMP2 and MMP14 increase, we found by performing a zymogram assay using the gelatin as the substrate, more active form of MMP9 on purple hyperoxia condition. This active form was absent on purple mice lacking neutrophil, suggesting that this activity was only dependent of neutrophil. So these two observations are consistent with their role in the tumor cell killing, which is highly manifested under hyperoxia exposition. We also found an increase of genes related to the generation of a peptide MHC class II complex and co-stimulation, this bona fide hybrid TAN. Importantly, a previous study has shown that hypoxic culture of tissue shown that this hybrid TAN generation was prevented by tumor hypoxia. Here we showed that relieving tumor hypoxia will increase likely the generation of this hybrid TAN. Among the most downregulated genes, we found that Siglec-F, which is a marker of a tumor-promoting subset in the lung cancer, was decreased. We didn't find any transcript of neutrophil elastase, which is consistent with a transcription of this gene during granulopoiesis. However, by performing a zymogram assay, by this time the casein as a substrate, we found that neutrophil elastase was decreased upon relief of tumor hypoxia, which is consistent with a reduction of tumor cell proliferation and its role described above. Capitalizing on this data, we performed the next flow cytometry on a PMN from a PMN tumor-bearing uterus, from tumor-bearing uterus and blood of purple mice housed in either in normoxia and hyperoxia condition. As a point of comparison, we also assessed for the phenotype of recruited PMN from non-tumor-bearing uterus, so in non-tumor environment, non-hypoxic environment after intrauterine LPS stimulation. So first, we noticed that the ROS expression was increased on PMN from uteri of Hyperoxia purple mice, which is consistent with the RNA-seq data, but not on PMN recruited after LPS stimulation. Similarly, and consistent with the RNA-seq data, we also found a decrease in the frequency of PMN Siglec-F positive from the neutrophil from uteri of Hyperoxia purple mice. And similarly, we didn't find any change in the frequency of the Siglec-F PMN on recruited PMN in a non-tumor-bearing uterus. Importantly, we didn't find any expression of ROS in the blood of PMN on each condition. We found 20% of neutrophil in the blood expressed Siglec-F. However, in the blood, we didn't find any change in Siglec-F expression, suggesting that the PMN phenotype was altered, and this phenotype was altered in situ within the tumor microenvironment by the tumor hypoxia. Moreover, what we found is PMN from uteri in recruited PMN and in tumor-bearing uterus can be divided in two different subtypes according to the CD11b expression. This difference is not apparent in the blood. Indeed, we found that 80% of PMN from uteri were CD11b low, and 20% of PMN from uteri were CD11b high. The frequency of this change remained unchanged by tumor hypoxia. We found that Siglec-F and ROS expression were only affected on the CD11b low subset. Indeed, as you can see here on purple hyperoxia condition, the frequency of Siglec-F positive neutrophil was decreased as it increased the frequency of ROS positive neutrophil as it increased their tumoricidal potential. With a similar analysis, we found that the class 2 expression, which corresponds to the bona fide hybrid tumor-associated neutrophil, was increased this time only on neutrophil from purple mice in hyperoxia condition in the CD11b subset. So as a conclusion here of this study, we found that neutrophil, as I mentioned before, shared both anti-tumor and pro-tumor activity. What we found is that tumor hypoxia will promote the PMN recruitment to the purple uterus, but will also prevent the cells to reach their maximum tumor combating potential by altering the PMN genetic program, the PMN functional product within the tumor microenvironment, will affect the phenotype of neutrophil. Relieving tumor hypoxia will compensate the associated reduction in PMN density. Indeed, relief of tumor hypoxia will control the PMN phenotype in this model, and as well as the PMN key effector molecule to improve their capacity to directly eliminate tumor cells independently of T cells. So as I mentioned, tumor hypoxia controls the PMN phenotype and key effector molecules. However, the mechanism through which tumor hypoxia controls directly or indirectly the conversion of neutrophil toward a pro-tumorigenic or an anti-tumorigenic phenotype remain not yet clearly assessed, and this is our first axis of current research. The second axis of our research consists to have a deeper understanding of the PMN product, of the role of PMN product and subset in tumorigenesis and in immunoediting, but I won't talk about this part today. So going back now to our main objective, which is to determine whether relief of tumor hypoxia is enough by itself to promote the conversion from an immunosuppressive tumor microenvironment to an immunosuppressive, permissive, tumor microenvironment. More importantly, our question is to see whether tumor hypoxia with the use of the respiratory hyperoxia or through the use of hypoxia-targeted small molecule are enough by themselves, or can be used as an alternative strategy to the current T-cell-based immunotherapy. So for that, and this is our third axis of research, we are now investigating how relief of tumor hypoxia affect or control the immune response in different human cancer, where this time first neutrophils are believed to promote tumorigenesis, and where this time T cells are known to be crucial to combat cancer. So first we want to determine how relief of tumor hypoxia steers the differentiation of PMN toward a pro or anti-tumorigenic phenotype. This role can be indirect first, and may involve the tumor epithelial cells. We have evidence that relief of tumor hypoxia directly affects tumor epithelial cells, independently of the PMN-induced response. Indeed, I showed you before that relieving tumor hypoxia massively decreased HIF-1 alpha stabilization. We also found that STAT3 phosphorylation was also massively decreased upon relief of tumor hypoxia, whereas the NF-kappa B inflammatory pathway remained unchanged. So the question now is to understand how this change affects the PMN phenotype. We already generated purple HIF-1 alpha logs mice. So on this mice, only the HIF-1 signaling pathway will be affected on epithelial tumor cells. And we found that this defect was not sufficient to induce any change in the PMN phenotype. We are now performing an RNA-seq analysis of sorted epithelial cells from purple CSF3 deficient mice to identify all the PMN-independent, hypoxia-dependent change variation in epithelial cells, and the change which are STAT3 dependent or independent upon relief of tumor hypoxia. Another possibility supporting an indirect role, it's a role for the stromal cells. We now exclude a role of leukocytes, and we are now looking for the non-leukocyte compartment by trying to identify any variation in endothelial cells, in the extracellular matrix remodeling, in fibroblast activation, or trying to identify any change in angiogenesis. The preliminary experiment don't show actually any change in angiogenesis, in the number of blood vessels, maybe in the size, but not in the number. Another possibility is that tumor hypoxia directly control the PMN phenotype. So for that, we are generating mice where this time, purple mice, where this time only neutrophil will be deficient for HIF signaling. So from these mice, purple mice mutated for HIF signaling in neutrophil, we will house them in hyperoxia and normoxia condition with the hypothesis that if there is a direct role, then the phenotype of this neutrophil, regardless of the hyperoxia or normoxia housing, will be comparable to the pro-tumoricidal phenotype than the neutrophil in the purple in hyperoxia housing. Lastly, we want to determine whether relief of tumor hypoxia could be used as a universal strategy to fight cancer. So for that, we are now investigating how tumor hypoxia control immune response in lung, prostate, colorectal, and breast cancer, which are cancer in which neutrophils are known to play a tumor promoting role and where T cells are known to combat the cancer. And we are also investigating how tumor hypoxia affects this time the immune response at later stage of the tumor progression. For all of this cancer, we use always the same strategy, always the same matrix, in which we are studying the effect of tumor hypoxia in model where PMN are present with or without genetic lesion, where neutrophils are absent, and in which case, in all of this case, with or without T-cell response. With the housing in normoxia and hyperoxia condition, we expect to reveal likely the hypoxia-dependent mechanism controlling directly or indirectly both PMN response and T-cell immunity. So 2 examples of our current results. We already found that neutrophil and T-cells are still required at late stage of the purple tumor progression, but this time by limiting metastasis. The metastasis in this model is reflected by an increased myoinvasion. The myoinvasion, it's an increase of epithelial tumor cells to the myometrium, which is the external layer of the uterus. We found indeed that there was less myo-invaded area in the regular purple mice with or without neutrophil, with T cells compared to mice without neutrophil or without T cells. And importantly, we already found that the hyperoxia reduced this myo-invasion. So the question we are trying to answer now is if there is a direct role, like at early stage of development of neutrophil, or if this time there is a direct role of T-cell immunity or an indirect role. In that case, this indirect role could involve the class 2 PMN, a subset of neutrophil, which is the frequency of this subset is increased upon relief of tumor hypoxia and can foster the T-cell response as it is suggested in lung cancer, in human lung cancer. And just a last example to show you that I won't show you all the results here, but we found on the colorectal cancer that the use of respiratory hyperoxia reduced by 70% in average the tumor volume in this context of colorectal cancer. I won't show you this result here, but we have evidence that neutrophils are involved in this process, and we try to understand now the role of each derived product. We also have an idea that there is an improved T-cell response, and we try now to understand what is this improved T-cell response under the respiratory hyperoxia, which can be revealed through a better infiltration of cytotoxic T cells, through a higher production of interferon gamma, granzyme B, or perforin, through an decrease of exhaustion marker at the surface of T cells, such as the TIM3, CTLA4, PD-1, a variation in Treg frequency, or any change in Trh polarization with, for example, a decrease in Th17 polarization. The last idea is to see whether relieving tumor hypoxia is enough by itself, or it can be used in adjunct of the current T-cell based therapy. So to do that, we can perform the same hyperoxia experiment with the adjunct, for example, of anti-CTLA4 or anti-PD-1 antibodies, and see if the use of the respiratory hyperoxia can circumvent the current limit of the based immunotherapy. So I will stop here for today. First, I want to thank Adrian Erlebacher for giving me the opportunity to work in the cancer immunology and drive all of this project. And I want also to thank all of the current member and past member at UCSF or in NYU. So thank you for your attention, and I will be happy to answer or trying to answer to your questions if any. So thanks for your attention.
All right, thank you very much, Karim, for this very exciting presentation. So with that, we're gonna go ahead with some of the questions. So we received a question, and so the first one is there any reason to choose 60% of oxygen as a hyperoxia concentration in your model?
We choose 60% of oxygen because it has been shown in human, in other species such as dogs and rats, that there was no inflammatory response, and that this frequency can, this percentage can be also used in a human with no sign of inflammatory response. So that's why we choose this percentage, because it can also be used in human too. 90% will induce strong lung damages, so we don't want that. So we want to see whether relief of tumor hypoxia can a good strategy. So the first point is to make sure that there is no inflammation in other tissue.
All right, great. So another question is, how would you apply this respiratory hyperoxia to human? And also, have you thought of other approaches, maybe using microparticles to deliver oxygen within the tumor microenvironment? And how do you think this will work in reducing tumor burden?
The use of respiratory hyperoxia by itself, you can use, the same kind of environmental chamber. Actually, this is what I tried to explain before. The HBOT, the hyperbaric oxygen therapy, it's a giant chamber where you can put a human inside. But in this case, there is an alternance of normoxia and hyperoxia at 90% oxygen, which is not what we want. So you can imagine a system where you can make people breathe this 60% oxygen for a few times. So this is the first idea. But of course, there is this better possibility to use hypoxia targeted small molecules. But actually, it exists. There is a company in South San Francisco trying to develop this strategy. I don't know yet the results with this strategy, but to develop that, it's important to decipher first if the relief of tumor hypoxia directly affect the tumor microenvironment to target, to identify new targets, or if it will affect directly leukocytes. So to develop the small particle, it's important to know what to target first. And that's why we try to understand if the relief of tumor hypoxia affect directly or indirectly the immune response.
All right, thank you. So then also we have another question. So could trained immunity play a role in PMN modulated cancer implication or therapies?
I'm sorry, I didn't understand your question.
So it's could trained immunity, so kind of like a trained the neutrophil modulated cancer. So to see, can we train those cells to really have them play a role in the cancer therapy?
Oh, you mean, for example, engineer neutrophil? This is what you mean?
Well, I don't know. It's really like how can you use those cells to really target the cancer and maybe reduce the tumor burden. You know, so instead of instead of you targeting T cell, how can we really target the PMN?
For example, you know, neutrophil can induce an immunosuppressive microenvironment by releasing the neutrophil elastase to inhibit the T cell response. Okay, what we found here is that relieving tumor hypoxia decrease the neutrophil elastase activity, so likely reduce the generation of an immunosuppressive microenvironment. This is actually maybe what happened with the use of the TH302 treatment, because they shown that they decrease the severe hypoxic area, but maybe in fact what they are doing is using this system is that they are decreasing the neutrophil elastase activity, which is required to maintain the severe hypoxic area. With this system, you can imagine that relieving tumor hypoxia will decrease the neutrophil elastase activity, thus improving the T cell response.
All right, great. All right, thank you. So I think we're running out of time because I think it's done. So I would like to thank everyone to be part of this presentation. And again, thank you, Karim, for this very interesting presentation on tumor hypoxia, which I think is a very interesting future direction in treating cancer. So with that, I would like to close the talk. And again, thank you everybody for participating to this interesting talks. And please follow the website for other interesting talk in the future. So yeah, thank you all. And
thank you for your attention. Thank you.
Thank you very much, Karim. Talk to you soon.
Bye.
Bye.